Hello, everybody. My name is Marcos Edgar Herkenhoff, and I'm a postdoctoral fellow at the University of São Paulo in Brazil. I will present the work entitled The Combination of Omics Strategy to Evaluate Starter in Probiotic Strains in the Catarina Sour Brazilian Style Beer, which was founded by FAPES. I would like to thank everyone for their attention, especially for being nominated. This study was recently published in the Food Research International Journal, where you can also find a version of this article for further clarification. Any question, you can also contact my email that I will leave it at the end. This work was carried out at the University of São Paulo in partnership with Food Research Center and with the Technical University of Applied Science Bureau in Germany. Let's go down to business then. Let's talk about beer. The consumption of craft beer is increasing worldwide. This also helps awake interest in beer with different flavors and the style, which includes and highlights sour beer. Thus, there are currently several breweries in tradition that are major, such as Australian and Italy, which have developed their own styles. Japan developed hop strains with special characteristics, and the Brazil is now with their own style of beer using fruits. Catarina Sour Beer is the first internationally recognized Brazilian sour style. Different from other beer, the beer manufacturing process has an additional lactic and fermentation step prior to the alcoholic fermentation by yeast. This confers an excellent flavor with pH 3 to 3.9, compared to conventional beers with pH of 4.2. This beer has been conquering the market, and currently there are several breweries that make it with the most varied range of fruits. As it is a beer reflective fermentation, this made us wonder. If lactobacillus is used to brew Catarina Sour, why not use probiotic strains? Probiotic strains can be defined as live organisms and microorganisms which, when administered at adequate amounts, confer health benefits of the host. There are several lactic acid bacteria species recognized as probiotics. Thus, probiotic lactic acid bacteria could be used to achieve the sensorial features of sour beer, especially Catarina Sour, which might also lead to a potential probiotic beverage. So this study aims to develop and evaluate the potential of the Catarina Sour beer style probiotic beverage. To test the survival of this bacteria, analysis was performed by microbiological growth by per plate and PM-qPCR. However, beer is also an hospital environment for a number of bacteria. Several factors mainly found in beer, such as iso-alpha acids, ethanol, low pH, high CO2, and low O2 may threaten survival, as well as temperature variation during the fermentation process. Despite this very important by hops, bacterial contamination in beer is quite common. The resistance that hops bring is related to action of specific genes, also negatively related to survival of bacteria in beer. They may be genetic markers for selection of probiotics to be used in beer. These hop-resistant genes marks have been identified in several microorganisms. So, thus this gene expression of the HOR-A, HOR-B, HOR-C, HA-B, S, R-A, and REC-A genes were evaluated by RT-qPCR. The liquid chromatographic tandem mass spectrometry, LC-MS technique also used to investigate protein that may contribute to the survival of bacteria and promotes positive interaction with yeast in the first moments of fermentation, day 1 and 2, which are the periods of adaptation of the strain. Metabolites, odor, and flavor were detected by headspace solid phase microextraction HSPMA, coupled with the gas chromatographic mass spectrometry GCMS analysis, with the aim to evaluate the yeast-bacterial relationship in the production of metabolites and compounds related to the sensory profile and metabolic pathway in the first 2 days of fermentation. The probiotic strains Lacticase Bacillus paracase subsp. Paracas F90 and 431 in the starter culture Streptococcus thermophilus TH4. And the three strains from Christian Hansen were selected for application in our beer formulations based on their abilities and survival in other food methods. In association with the yeast, Saccharomyces cerevisiae strain Hep Ale US Nu 5, due to its characteristic, due to its characteristics. The Catarina sour formulations were designed by Beersmith software. 15 formulations of Catarina sour evaluated. 1 liter of each formulation was brewed in duplicate to avoid variation in the wort composition. A single batch of 30 liters of wort was meshed in distributed formulation. Brewers divided in 3 steps. First, 50 degrees Celsius degrees for 12 minutes. Second step, 72 Celsius degree for 60 minutes. Third step, 76 Celsius degree for 10 minutes. After mashing, the wort was filtered and boiled with pulps in a boiled tank. All formulations were evaluated using the following steps: microbiological count, microbiological count by pure plate and PM qPCR by using ABI real-time system, the viability of the special profiles of gene use also a real-time system. PME is intercalling substance that intercalate with the DNA, and so you are after this reaction, the DNA from depth cells are missing, so you can doing precisely count. The both gene expression and PM qPCR was performed using CyberGreen. LS-MS for proteome analysis and HSSPMA GCMS for metabolic and flavor components analysis, as I said before. So, plate count and PMA qPCR show different counts by per plate, counts ranging from 3 to 7.3 logs PFU per ml, while the PMA qPCR ranging from 5 to 9.90 to 8.77 after 1 month of storage at 4 degrees Celsius. As you can see, comparing the left and right columns, PMA qPCR has a height count. Beer's stressful metrics for bacteria, which explain that lower counts were obtained from per plate counts compared to PMA qPCR. Although the expression of these resistant genes, especially here HOR-A, HOR-C, and HIT-A, are associated with these strains, as studies by other authors have shown, as well as in our data, they make some contribution to spoilage or hope resistance in beer. However, the evidence points out that they are not fully determined. As reported previously, our data highlight HOR-C and HIT-A. HOR-C genes seems to be involved with microbiome resistance in control and passion fruit juice, while HIT-A in formulation with peach juice. Furthermore, there were different protein pathways in the network performed by string software and U in the yeast. Now, formulation they showed not network involving glycolysis, the protein network related to carbohydrate metabolism were showing in TH4 passion fruit, TH4 peach, F90 control, F90 passion fruit, F90 peach, L4 431 control, L431 passion fruit and peach. TH4 peach, F9 control, and L431 passion fruit and peach show at the lowest counter networks of interest, how you can see in this graph, in these figures. Regarding the protein networks of TH4, F90, and L431, TH4 showed interaction networks in all samples, the low interaction in all samples compared to F90, L431, as you can see. This was already observed on day 1 when yeast strains was absent. In contrast, F90 and L431 showed an increase in the number of proteins in a regimen in functional enrichment at day 2 in virtual formulations compared to day 1. In addition, the presence of proteins involved in network related to the response of stress in glycolysis was observed in F90 and L431 strains in addition to glycolysis related networks. Cell adhesion proteins could also be evidenced. The DNA binding protein, which is widely found in F90 and L431, could be highlighted. Comparing to the compounds identified by GCMS, in the formulation with TH4, 27 compounds were found that are from the yeast database, and therefore they are not naturally produced by yeast. Meanwhile, 28 were found in the formulation with F90 and 35 in the formulations with L431. Among these compounds, 70 were produced in formulations with TH4 or F90 or L431. Two compounds known as 2-ol and dodecanol were found produced in formulations with TH4 or L431. So, in conclusions, our data corroborated the fact that probiotics, especially F90 and 431, may be the best candidates for the use in breweries, as they may have several defense mechanisms such as membrane adhesion proteins and H-pump. Our formulations showed a probiotic viability between 5 and 8 logs equivalents throughout the manufacturing stage. In addition to the expression of the C gene seems to be involved in microbiome resistant control passion fruit juice added formulation, while HA was only important in the formulations with peach. However, as several other studies also suggest these genes may not be determined in microbial survival in the beer. Proteome analysis indicates that strains L431 may be the one of the best adapters to beer conditions, and flavor analysis indicates that the strain used the interfere with flavor and odor. So I'd like to thank my team at the University of São Paulo, some of my team in TH Bildo in Germany, Christian Hunter for making the probiotic strains available in the ecology of beer and malt in Blumenau for technical support. Thank you for your attention.